So, we've all heard about the Pfizer and Moderna mRNA vaccines. For the past few months, I've been hearing about it, but I've been busy helping with another COVID vaccine that uses a different mechanism, so I didn't have the time or headspace to look into it. I was finally able to sit down and look at it over the last few weeks, and holy crap, they weren't kidding. mRNA vaccine technology is a miracle medical wonder that can be used in so many different ways it's ridiculous. It's been a holy grail for decades, but always gave severe immune response reactions, so it was a no-go, and was left to the lesser-sung scientists to do the research on it. While places like Moderna were getting close to figuring it out over the last 10 years, they probably assumed it would take another 5 to 10 years to reach clinical trials and be used in people. Then COVID hit and the money was poured into all the most promising vaccine technologies, mRNA being one, and they were able to finish up their work quickly and jump directly into clinical trials. Pfizer was the first to get the results, but Moderna had the advantage of the mRNA having an oily coating that allowed it to be stored at minus 20 degrees, as opposed to Pfizer, which requires a minus 80 degree freezer. Usually this kind of technological leap requires a war, but a lockdown of the world's economy was good enough in this case. We currently have a shortage of minus 80 freezers for commercial use, which is funny because I worked with minus 80 freezers for most of my career and just assumed they were standard in places like hospitals. It makes sense after thinking about it though. You may have heard headlines about mRNA being a huge breakthrough that can change science, but if you aren't a scientist, what they say may not make much sense as it's complicated. So I figured I'd start by breaking that down. So strap in. This is all understandable, but there's a lot of moving parts. It's kind of weird to actually discuss something in my field as opposed to stuff I picked up along the way. So here we go. Starting out, what the heck is mRNA? Let's tackle that by doing some basic biological definitions. RNA stands for ribonucleic acid. It's found in all living things. There are even viruses made out of it. It has many shapes and can fold into many things. However, it has a flaw. It's fragile and can damage easy, which is actually useful, but also why it's not a good long-term storage medium for a genetic code. DNA stands for deoxyribonucleic acid. It is missing an oxygen on the backbone subunits, making it much more stable and less prone to damage or mutation. DNA is usually in a double helix, you most often see, but can fold into other shapes. I won't go into deep detail as there's so much to this, but as you know, our genetic code is made up of 4 base pairs. They are adenine, thymine, guanine, and cytosine. Thanks to chemical forces, adenine and thymine stick together like magnets, and so do guanine and cytosine. So every forward code has a negative code using its polar opposites. Polymerase is a molecular machine that unzips the DNA strand at the right place and begins taking base pairs and sticks them to the coding regions so it mirrors what is on the coding strand. DNA polymerase duplicates the DNA using DNA base pairs, and RNA polymerase does what is called transcription, and only converts a segment of DNA into RNA. However, in this case, just due to chemical reasons, uracil takes the place of thymine in RNA. The polymerase can read transcription start sites made of specific combinations of base pairs, so it won't start coding until it finds that particular sequence. Once the stop sequence is found, the polymerase releases the RNA strand and lets it go floating away. The polymerase continues to read DNA until it finds the next start sequence. So the RNA chain is now floating around in the cells. For the sake of our discussion, and because I haven't studied it in about 10 years and I know new findings have come out, it can do 3 things. It can do more, but that's beyond the scope. It can become mRNA, tRNA, or rRNA. There's also ribozymes, but that's its own subject. rRNA self-folds using many of the same chemical forces that allow the DNA base pairs to stick together, to make up a subunit of a ribosome, which does the same thing that polymerase does, but instead of converting DNA into RNA, it converts RNA into a string of proteins. tRNA is transfer RNA, which creates lock and key mechanisms attaching to 20 specific amino acids and having 3 points that match with specific RNA base pairs. The mRNA or messenger RNA is found free-floating by the ribosome, and the ribosome will start reading the mRNA. The ribosome then reads the first 3 base pairs called a codon in the chain and takes the tRNA that matches it and attaches an amino acid to the chain and continues reading 3 at a time until the mRNA chain runs out, at which enzymes will chew up that mRNA. As you can see here, the codons align with the amino acids, and some amino acids have duplicate codons, which is useful in the case of mutation as it has a higher chance of making these codons with little changes at all. Okay, so big overhead view. DNA goes to RNA, goes to amino acid chain. These chains then self-fold just like the rRNA, just based on each amino acid's chemical bonding properties, and goes on to make up every part of you. Some fold into nanomachines, some into structures, and some into other functions. This is called the central dogma of genetics, though HIV made the world aware that there is an enzyme called reverse transcriptase that can convert RNA back into DNA, which is what HIV does. HIV is made of RNA, and once it enters a cell, it bumps into ribosomes that will read and code it. From this, reverse transcriptase and integrase are assembled, and the reverse transcriptase then takes the RNA virus particles and converts it to DNA. The integrase then inserts the DNA into the cell's DNA strand, so the polymerase will read it later on, causing the virus to duplicate itself. It can either quickly reproduce and kill the cell if the immune system is weak, releasing its virus particles, or it can just hang out hiding until it detects weakness, then kick in mass reproduction. This is why HIV is so crazy deadly. Antiretrovirals block reverse transcriptase from working, so it stops RNA from getting converted to DNA. This gives the immune system time to fight back and kill off infected cells and floating viruses if taken prophylactically, or slows the propagation of HIV until the body gets too weak, these days with old age, but in the past, pretty much any infection. Anyway, sorry tangent, but I use that to help explain where mRNA vaccine comes in. We can now synthesize mRNA of our own to hypothetically do anything we want. The special coating on it helps protect it from enzymes in the blood and helps it enter immune cells and change them to tell them specifically to target COVID. It does this by having the ribosome read it and produce a spike protein. Upon building the spike protein, which is an inert but identifying part of the virus in question, the protein activates the immune system and starts reproducing immune cells to specifically target and kill anything with that spike protein on it. This is groundbreaking. For the longest time, our only tool for vaccinating people involved growing up the live virus and weakening it or killing it, or in more recent years, growing up a genetically stripped down virus of another species with only the antigen or particles of the virus in question that the immune system could use to recognize infecting agents. This skips all of that growing and targeting period and jumps directly to the body making its own spike protein because we gave it the instructions to do so. With this tech lined up and any new virus or bacterial pandemic that comes along, we could respond even quicker than this time around, as once we've identified target proteins, we can just immediately jump into clinical trials and production. But it has even more potential in cancer than even in pathogen response. The most groundbreaking cancer treatment of the past decade has been CAR-T, or chimeric antigen receptor T-cell, made most famous by Jimmy Carter, a man in his late 80s having melanoma that migrated and infected to his liver and brain, and he beat it, coming back healthy, something unheard of just a decade ago. The process is amazing but very complicated and also expensive. They take your blood and then put it through a process that filters out the right immune T cells needed. They take these cells and then hit them with the genetically stripped down virus that introduces genes that activate these cells to identify and attack those specific cancer cells. They then grow up the cells to a certain level and then re-inject your own modified immune cells back into you, which makes your immune system start making these cells in overdrive, directly attacking and killing all of that kind of cancer cell. This process works, but is complicated and expensive as it requires transporting the blood to a sterile, heavily controlled facility, and multiple chains of actions that could lead to contamination, and QC needed to ensure it worked and has no contamination, and then sterile transport back to the doctor and patient, and sterile injections needed. mRNA will skip all of that, allowing only a nurse or phlebotomist or pharmacist to just give you a shot, and the mRNA goes into your cells and kicks off the process. mRNA costs the same no matter what you encode. The CAR-T industry and investment right now are going gangbusters as there are several hundred different cancers based on cell type, and each company has their own specialty they're investing in. This will make cancer treatments in the future dirt cheap, and in about 20 years, as cancer detection becomes more and more sensitive, you could go to the doctor and get a simple blood test. The doctor may tell you that you have very early cancer of a specific type, or you're genetically predisposed to a certain cancer. then just vaccinate you for these cancers before they ever have a chance to get a foothold, and you'll never have to worry about them other than maybe a booster shot. They may also give you a vaccination against cancers based on your age, based on past data, and if they think you just need a booster based on studies. In 20 to 30 years, cancer could very well be just as annoying and expensive to treat as the flu, having to get a different variant every year. This will drastically extend life expectancy if we can get other issues under control. On top of that, mRNA can code for CRISPR machinery that allows cells to modify their own DNA, allowing for one to genetically alter their own cells at any time. At first, of course, we'll be targeting debilitating genetic diseases, blocking certain chemicals made, such as in Down syndrome, or adding chemicals needed that the average person naturally makes. After that, we'll target immune issues like food allergies and asthma. After that, who knows, they could allow for cosmetic genetic modification, using mRNA to change metabolism, susceptibility to hunger, eye color, hair color and type. They could regrow hair. The lists are endless of the craziness we could get after all the major genetic and immune issues are fixed. On top of that, life extension. Telomeres are sequences at the end of chromosomes in your DNA. After being replicated enough times, they stop being able to reproduce. This is believed to have a big impact on how aging works. After a certain point, your cells just expire and stop replacing themselves. One could take telomerase, which will put extra telomeres on the end of your chromosomes, but it's also highly carcinogenic. However, once cancer is just an annoyance, it won't be that big of a deal, and perhaps mRNA may be able to add telomeres without increasing the risks of cancer. Also, it's been found that as one ages, their blood at a certain point actually produces low-level toxins that inhibit repair and the immune system, which is why Silicon Valley is in a crazy fad right now to buy poor young people's blood and put it into rich old people. mRNA could tell blood cells to stop making these aging factors and give an older person the blood of a young person. After you turn 30, the ratio of blast cells, which build things, and clast cells, which break things down, start to inverse, causing healing to slow down in this way as well. mRNA could kick in blast cell production to return to pre-30s level, which could not only slow or stop aging, but completely reverse it. After that, we would just need to perfect 3D organ and tissue printing for organs that I've discussed in a previous video that will just wear out like kidneys, though mRNA may make the speed of replacement needed much longer. Then we need nanobots to clean up and purge our bodies of the heavy materials we build up and acquire over time in this polluted environment, and as far as we know, there's nothing aside from accidents or inadequate access to these treatments that could kill us. And even if there are more factors involved that we're unaware of until we clean out all of these other problems, it will still radically extend a person's life. Now, using mRNA for intercellular CRISPR may take much longer than vaccinating against most diseases, cancers, and allergens, as there may be other issues we may be unaware of, just like genetic engineering was all the hype in the 90s that we discovered was way harder than we expected, but just being able to beat cancers, pathogens, and allergens would raise the standard of living and lifespans of people worldwide. So yes, this is why the mRNA vaccine is such a big deal. We have just started in this tech, and once scaled up, it will just get cheaper and cheaper. For many diseases and cancers, we already know the gene sequence needed for vaccines in CAR T we currently use, and converting them to mRNA would take little to no effort. After that, it's just a lot of money for clinical trials, and then there will be vaccination specialists that keep banks of different mRNA vaccines near you may go to yearly to maintain your own body. Right now, COVID sucks, but our offspring and many of our future selves will be grateful that this virus lit a fire under the government's asses to fund this research that may have taken a decade longer that will impact and improve our lives in the centuries to come. Of course, we could just vote for politicians who believe in and fund science at the level we used to fund NASA, but that would be silly. We need to use that money on war and making rich people more money. Once this tech is available, we will now have to figure out how to make a world that people would actually want to live in for more than the 100 years they have now. And that may be even harder work than all the work put into it making this technology, but we shall see.